Hello, good morning. Today we are going to see about types of RNA. We have seen about RNA, but now we are going to see about types of RNA. There are three types of RNA. One is mRNA. mRNA means messenger RNA. And this mRNA is produced from the DNA, right? And tRNA transport RNA, rRNA. Okay, there are three types of RNA produced from DNA. And these three RNA, this messenger RNA, brings the codes. It brings the codes, okay, acts as a template. And the tRNA transport amino acids. Transport amino acids. And a ribosomal RNA is synthetic in function because it has around 100 different enzymes, so which helps in bringing this amino acid combined with one another and makes a long chain of proteins. These are the three types of RNA. mRNA, it acts as a template which has codes. tRNA, transport RNA, rRNA, ribosomal RNA. Now we are going to see about the transcription process. The process of transcription. Okay, first we see in a lower animal, bacteria, how it takes place in bacteria. Then we will see in a higher organism. So, in the process of transcription, formation of mRNA. So, transcription is formation of mRNA. What is the most important is RNA polymerase, an enzyme, which is the basic factor which helps in the process of transcription. In bacteria, this RNA polymerase did the work of catalytic work, and it does all three steps. What are the all three steps? 1, 2 and 3. It initiation, elongation, termination. So all three steps: initiation of the process, elongation of the process, and termination, ending the process. All these three are done by this single RNA in bacteria. But whereas in eukaryotic cells it is different, right? So we are going to see how this initiation, elongation and termination takes place. Okay, these three steps we are going to see. The first one is initiation. how it get initiated. See, the RNA polymerase, because it is involved in all the process, okay, it joined with promoter, okay, it is an initiator, it joined with the initiator, and that initiation factor is called sigma factor. This is called the sigma. So when it joined with the sigma factor, it become an initiation. It initiate the process. And while joining with this, it takes NTP, like ATP, nucleoside triphosphate. It's an energy compound. It takes energy from nucleoside triphosphate and join with an initiation factor. Now this is now ready to start the process of formation of mRNA. So it initiate the process. I am going to explain with the diagram, right? So this is initiation, okay? Then once the initiation is started, the next stage is elongation. The elongation starts, and it started moving, forming the mRNA, right? Once the elongation is over, at the third part, termination. Now this RNA polymerase joined with the termination factor. There is another factor called Rho. The name is Rho, but mentioned in the letter P. Rho factor. When it joined with the Rho factor, the process stops. Only 3 steps. Very simple step. RNA polymerase joined with an initiation factor and start the initiation of the formation of mRNA. For that, it uses nucleoside triphosphate as an energy molecule. Once it forms the initiation factor, what it does? It breaks the helix. That's the first step we have seen. It breaks the double helix, then the process starts. Then the continuation of the formation of mRNA, then the termination. If it join with the factor, Rho factor. And this is a very simple step in bacteria, right? I am going to explain this with a diagram, right? So 3 diagrams I will do, right? Okay, this is 3, this is 5. So this is 3 and this is 5. Because you see, 3, no, that means this is 5 and this is 3. Okay, this is how the DNA is. Now we are going to see how the initiation, the first step takes place. Okay, I will just draw here an RNA polymerase. This is RNA polymerase. RNA polymerase. It is connected with a factor, sigma factor. Now it's connected with the sigma factor. Now this is initiation. This is initiation. Step 1. Initiation started. Once the initiation started, what happened is Now this part of it connects, and the process of transcription starts. This is the first diagram which you have to draw. The second is elongation. What happens in elongation? We will see. The same diagram. Okay. Instead of here, I'll make it here a little bit. Okay, now this is elongation. Now it has started moving towards this direction. Okay, now this factor is here. This factor is here. And what happened to the transcription, which I have shown in, this is mRNA actually, formation of mRNA. Now this mRNA is formed like this. Long, it is getting elongated. It's a small piece, now it started elongated like this. So elongation continues. And in the third stage, termination, end process. In the end process, the same diagram, I will just, this is very difficult to draw here, I will use the same diagram, I will shift this now, I will make it here. Okay, now I will change this diagram. Now it is here. This factor is here. Now you join with row factor. Okay? When you join with the row factor, make it in a different color. And if you join with the row factor, then so now the mRNA is broken because of a different color I have given, which is in a green color. This is mRNA. Is released out. Okay, these are the three steps involved in bacteria. You have to draw this diagram. The sigma factor with the RNA polymerase, it starts initiation, the mRNA formed here. Then it get elongated. So in between you draw this, make a little bit longer. Suppose this one. Now this is in the second stage, this is in the third stage. mRNA is totally formed. Now a small piece now become elongation, and third one is termination, where it is joined with a different factor called row factor. When it joins with the row factor, it breaks and releases the mRNA. This is the process which takes place in bacteria. It's a very simple 3 steps. On all these 3 steps are taken care by one single mRNA polymerase and enzyme. Okay, now we move on to the next.